trial.
Thank you. This is only the ongoing study, so I only introduced the ongoing study. This is my SUA disclosure. This is, as you know, very famous algorithm, clinical practice guideline, and not only USA and Europe and Japan is also included. That is now renewed by Dr. Kasper. This published in German Oncology. The patient referred to sarcoma referral center, and tumor was in the situation of tumor was threatening to life function and QOL. No. The answer is no. Frontline approach is active surveillance, and the answer is yes. The active treatment is necessary. About the extremity and the girdle and chest wall desmoid tumors, primary active treatment is medical therapy or sometimes ablation. But in Japan, ablation is not allowed so far. But most of the available active treatment, medical therapy, at least 6 months is required to see the efficacy. And about low-dose cytotoxic chemotherapy, like meso and bimbracine, longer duration may be required. This is CPG in Japan. Probably you can't read. You can read, okay. That is similar. That is similar to Europe and the USA. And active surveillance, not good. The active medical treatment or surgery in case of abdominal wall. In Japan now, meso-bimbracine can be used, and pazopanib and doxorubicin and dacarbazine can be used. But sorafenib and niro cannot. Please save the patients by SpringWorks, hopefully, in the near future. The side effects, sometimes side effects annoyed and high cost annoyed about the pazopanib. Some patients can't allow to pay the costs of pazopanib in Japan. So we think it is necessary to develop drug treatments that are effective and few side effects and inexpensive. We use the drug repositioning method using this human desmoid cell culture. We extracted these drugs, drug repositioning, and we chose auranofin because of the not poison and not can be used clinically. That is auranofin. And not only the in vitro, the in vivo, Dr. Ben introduced the APC, spontaneous onset desmoid mouse. He provided me the APC mutated mouse, and we use these mouse and administration of the oral administration or intraperitoneal injection. Both the onset of the desmoid is inhibited by auranofin. That is published in scientific report, and based on these results, we designed the single center, now university single arm open label study that is registered JRCT, Japan Registry of Clinical Trials. This is scheme of this trial. Initially, we planned registration cases, the number of cases, 23 cases, based on the statistical estimation. The primary endpoint is PFS at 26 weeks. These are secondary endpoints. It contained goddess that is translated into Japanese. The Murinar allow me to translate into Japanese. So we use this goddess also. 23 cases planned, but unfortunately, there are not PR cases, and fortunately, there is only one case of PD. So we decided to discontinue this clinical trial after inclusion of 16 cases. One case was excluded because of the initial inadequate laboratory data. So finally, we entered 15 cases. Now that we finished only the primary endpoint, 26 weeks PFS. This is demographic data. The mean age is a little bit older than the standard cohort of the desmoid, 49 years. The location is neck in 2 chest wall 3, abdominal wall 3. And mean maximum diameter of the tumor is 67 millimeter. The PFS at 26 weeks, the rate is 93%. It is not bad, but the other secondary endpoint is not evaluated yet. So next year, I will present the whole data. Whole results will be presented the next year. Thank you.
Thanks. So we're open for questions. Yeah, go ahead. Yeah. And make sure the microphone is on. So yeah. Good.
Patients enrolled in the study were progressing. Maybe I didn't see, or
Yeah.
kind of disease.
Yeah, it is a very good and tough question for me because of the single institution-based clinical trial. So progressive means not based on the RECIST, but physicians-based, physician and patient. So pain is worsening. The not RECIST, but the size is minimum growing. So that is the that strongly affect the result, I think. Yeah.
A question?
I think the mechanism of action might be
Auranofin? Yeah, that is analyzed in vitro and in vivo. The auranofin generally inhibit the malignant tumors through the mechanism of reactive oxygen species, but we check the effects of the auranofin to the cultured desmoid cell line, but no effect on the reactive oxygen species. But the beta wind catenin beta wind and beta catenin pathway, that auranofin generally is not reported to affect the wind beta catenin pathway, but based on our result of the in vitro, the downstream of the wind beta catenin pathway, like action 2, that is inhibited by auranofin. That is, I evaluated.
looking to see if you see evidence of that mechanism of action.
Uh, No. You mean the tumor?
Harvest some sample. Pull out the sample to see if you see
Oh,
mechanism being produced by
yeah. You mean that based on the non-clinical data, non-clinical data. So about the in vivo data, we don't have. In vitro data, as I said, the downstream of the genes is affected, but the beta-catenin location in the cells is not changed by the auranofin. So it may, I can't explain that the beta-catenin location is not changed, but probably auranofin affect the post TCF activity. That is the way I can only say based on my results.
How are the toxicities and tolerability?
Toxicity of the auranofin? Yeah, that is, we use a various concentration of the auranofin to the cells, not only the desmoid and but also the fibroblast, normal fibroblast. The concentration we used, no toxicity is observed. It is okay? My question answer is okay. Yeah.
Since you got that there is the a potential role of immune system of immune cells
You mean immune cell?
is active in arthritic rheumatoids. You got something with this? Am I correct?
Probably.
I can ask you later.